Perfect. So if you're just joining us, welcome. And if you've been listening to the chit chat, welcome again. I'm very pleased to introduce today's speaker, Dr. Marjorie Lundgren, who is a UKRI Future Leaders Fellow and lecturer in plant environmental physiology at Lancaster. So Marge is a fellow environmental ecophysiologist, and she really applies phylogeographic methods to understand diversity and how photosynthetic systems have evolved. And what we will hear today is, I think, the really exciting next stage of her research. And now how can we direct this and engineer C2 photosynthesis, which she'll explain, to enhance food security. So with that jotty introduction, Marge, please take it away. And we're all looking forward to hearing from you. So thanks. If you have questions, you can put them in the chat or save them to the end, and I'm sure we'll have time for them. But thanks very much.
Perfect. Thank you, Amanda. Right, so today I'm going to tell you about C2 photosynthesis broadly, and I'm going to tell you about my efforts to introduce it into crops for improved food security. You notice that I have a tentative kind of question mark at the end there, and that'll kind of keep coming up throughout the talk. Does it make sense to engineer C2 photosynthesis into crops? What are some of the pros and cons of this approach that we need to keep thinking about? I'm going to divide my talk into two parts. The first part is just background on C2 photosynthesis, kind of what it is, its role in C4 evolution, and how they interact with the environment. And then the second part is all this engineering work. Why do I think C2 photosynthesis has potential to improve crops? What kind of modifications would be required to recreate it in a C3 background? And what is my particular approach to this engineering effort? Okay, so starting with the part one, what is C2 photosynthesis? And before I get into that, I should highlight that it's also known as the photorespiratory CO2 pump or the glycine shuttle. I also like the fact that it's actually an oxymoron. Like saying C2 photosynthesis is just like saying photorespiration photosynthesis. So, it makes me chuckle every time. I know that we probably need to rethink the terminology here, but I've adopted this from some previous researchers, kind of like the Monson-Sage models. And so I'm keeping it up for now, but I realize that it can cause confusion saying C2 photosynthesis. So I'm going to clarify what specifically I mean when I talk about C2 photosynthesis in the next couple slides. Right. So C2 plants act just like C3 plants, and that they'll do the Calvin-Benson-Bassam cycle, or the Calvin cycle, in the mesophyll cells. But that's really just under non-photorespiratory conditions. And when you get under photorespiratory conditions, that's when you start seeing differences between C3 and C2 plants. And C2 plants will take up oxygen, and that'll kick off the process of photorespiration. And in a C3 plant, that happens entirely within the mesophyll cells. But in a C2 plant, they start the process of photorespiration in the mesophyll cells, but then the resulting glycine, it accumulates in the mesophyll cells. And the reason it accumulates there is because GDC, which is just glycine decarboxylase, in a C3 plant, that's just going to be in the mesophyll. So that's why you can do the whole photorespiratory process in the mesophyll. But in a C2 plant, it's over here, it's in the bundle sheath. And so that causes glycine to start accumulating in the mesophyll because it can't be decarboxylated. And that eventually there's a passive diffusion into the bundle sheath, where it can then come into contact with glycine decarboxylase, be decarboxylated, and that releases CO2 that can then be re-assimilated in a secondary Calvin cycle there. So essentially, C2 photosynthesis shuttles, concentrates, and re-assimilates photorespired CO2, making it a natural CO2 recycling mechanism and also a CO2 concentrating mechanism. I put up this other diagram. It's a bit better than my little diagram over here. This is from Schutler and Weber 2016 paper. And I just put this up real quick to remind me to say that when you decarboxylate the glycine, that also not only does it release CO2, but it releases ammonia and serine. And serine then goes on to create through PEGA in both cell types And also the ammonia then goes into this pyruvate cycle. And that's going to come up again in a couple slides. So I just wanted to quickly highlight that here. Okay, so what does C2 photosynthesis, what role does it play in C4 evolution? And I put up this slide. So this is this is the SAGE model of C4 evolution. This is the pyramid model, and I don't really love the directionality of it, but it's commonly used, and it'll demonstrate my point well enough. Which is, if you think of the transition, the evolutionary transition from the ancestral C3 state to the kind of the complex C4 state, which looks very heavenly up here up in the sky. Anything that falls in between those two kind of typical C3 and C4 phenotypes is going to be considered a C3, C4 intermediate. But then you can break that down into more steps. And within that C3, C4 intermediate, you'll see their protocrans, which are just kind of like an anatomical preconditioning step for the most part. And then this is where C2 photosynthesis comes into play here. And these C2 plants are very specifically those that are using this glycine shuttle that I talked about in the previous slides. And then when you get a bit closer to C4, you have these C4-like plants, which are running a good amount of the C4 cycle as well. So what I want you to take away from this is that C2 photosynthesis, or these C2 plants, are a subset of C3, C4 intermediates, because you'll see both of these terms in the literature. And a lot of times people use them interchangeably, and they're not exactly interchangeable, because C3, C4 intermediates is a broader term. C2 plants and C2 photosynthesis very specifically refers to these plants that are using this glycine shuttle. And that can actually be broken down one more step into this C2 type 1 and C2 type 2. And all that means is, in the type 1s, those are just using the glycine shuttle. But then you sometimes have individuals that use a glycine shuttle and a weak C4 cycle. So lots of physiological layering here. I think I found this other diagram. This is in another SAGE review. And I thought it kind of showed it maybe a bit better, where you have the gradient from, oh, can you see my pointer? Hopefully you can see my pointer. Okay, good. So we have the gradient from C3 all the way kind of down to the C4 state. And you can see where the C2 falls in that larger scheme, both type 1s and type 2s. Okay, so that's what I mean when I'm talking about C2 photosynthesis. So what role does that physiology play in terms of C4 emergence or C4 evolution? And there's a few key things that it's doing. First, C2 photosynthesis establishes this dual compartment system that's required for C4 photosynthesis, this mesophyll bundle sheath dual cell system. And for that to happen, these C2 plants must start initiating this improved connectivity between the two cell types. So that's one step closer to making C4 work. C2 plants also introduce the bundle sheath Calvin cycle. And in order for that to work, there must be bundle sheath organelle enrichment. So again, you're kind of stepping towards a C4 phenotype. There's CO2 concentrating mechanism. So the fact that these C2 plants have the concentrating mechanism and they recycle the photorespiratory CO2 means they can actually do better in high photorespiration environments, meaning they're improving the performance in new environments like warmer temperatures, for example. So basically what I'm trying to get at is they're shifting lineages into new environments that can then select for further emergence of the C4 phenotype. And finally, this last step, so there's a hypothesis out there that this shuttling of glycine into the bundle sheath, and it decarboxylates and it releases CO2, serine, and ammonia. That ammonia, it's believed, might build up in the bundle sheath, and that would cause an imbalance, this nitrogen imbalance between the bundle sheath and mesophyll cells. And one of the best ways to ameliorate that imbalance is the evolution of a C4 cycle. So the idea is that these C2 plants have this nitrogen imbalance, and that selects for C4. With that said, I'm adding these little question marks there because, I mean, if you follow the literature back, this has never actually been proven or shown, and I think it's just a hypothesis. And I think I've got a PhD student looking into this. I'll talk about that more in a minute, but I've got a different PhD student looking into this, and we think that the nitrogen might be able to be sorted out within each cell type. And we're working on this, and we're nowhere near being able to make any kind of strong statement. But my point is that, this nitrogen balance might not be the main selector for C4 photosynthesis. And if that's the case, there might be something else going on, and it might be really interesting. So we're working on that. So that's why I just put those question marks there, because it's something I think as a community we need to be a bit skeptical of until we can have a bit more proof. Right, and so I wanted to just quickly have a shout out for one of my PhD students. This is a different PhD student, Sophie Young. She's doing some very cool work trying to understand why C4 photosynthesis is so rare in trees. We know that C4 photosynthesis has repeatedly evolved something like nearly 70 times independently. If it's evolved so many times, why is it not happening in trees? There are a few true trees that have C4 photosynthesis, but the hypothesis is that they were already C4 lineages before they became trees. So could it be that once you're a tree, you can't then evolve C4? And the reason I'm mentioning all this now, besides that I want to give a shout out to my student, is that it could be that these evolutionary intermediate steps, this C2 phenotype, is the limiting factor in C4 emergence in trees. And to start determining if that's the case, there are a few tree species we think might be C2, and hopefully once she can start traveling again, we can confirm that. But yeah, so this is kind of an interesting little tangent to the C2 research, is its effect on C4 evolution in trees. Okay, so moving on, C2 photosynthesis is rare, right? So it's known to exist in fewer than 60 species currently. With that said, I do think it's far more common. It's a bit of a faff to characterize. If you look at delta 13C stable isotopes alone, these plants are often going to look like a C3 plant. And so I think a lot of species get swept under the rug and just get called C3 when in fact they probably could be running C2. I also think there's a lot of interspecific variation. So there could be that some populations are using C2, whereas other populations aren't. And if that's happening, then it'd be really easy to miscategorize a whole species as C3 when in fact there is C2 going on. So what I'm rambling about is just that I know that it's currently rare, and even if we find a lot more species, it will still be rare, but I think it's not this rare. Okay. And of all the species that we currently know that use C2 photosynthesis, the only one you've probably heard of is rocket, or arugula, as it's called in America, and it's Diplotaxis tenuifolia. Other than that, there's a couple kind of horticulturally used species, but none of them are really dominant species that you would know of, probably. Right, so despite its rarity, C2 taxa are incredibly geographically and ecologically widespread. Each of these green points is an occurrence data point where it's been found in the past. And you can see that C2 taxa have been found on every continent except Antarctica. And yeah, there are a few blank spots in sub-Saharan Africa and Asia, but I don't think they're absent because C2 plants aren't found there. I think it's just a collection bias because this is based on herbarium records. So point is, C2 plants are broadly distributed. The second point I want you to take away from this is that these plants can grow in extremely contrasting environments. So they're found in very hot places, like here in Pakistan. They're found in incredibly cold places, like in northern US and in Canada. They're also found in the Rocky Mountains, where there's dominating frost days per year. And importantly, they're also found in diverse precipitation environments. You can have C2 plants in areas that are incredibly wet and also incredibly dry, like this distinction here between the rainforests of Colombia and the coastal deserts of Peru. So point is, C2 plants are not only broadly distributed geographically, but environmentally, they can seemingly tolerate incredibly diverse environments. Right, and because of that diversity, ecological diversity, we think that they can both promote or limit C4 emergence. And we think once a C2 condition emerges, that shifts lineages into warmer, more seasonally wet environments. And once you're there in those kind of warm, seasonally wet environments, that then allows those lineages to there's selection pressure to then become C4, right? And that's some of the results we found in this paper, this JXBot paper I wrote with Pascal Antoine. This is 2017. But if you look specifically at those lineages, there's 5 lineages that never evolved C4 photosynthesis. They just evolved C2, and they seem to have just kind of stopped there, right? And these lineages live in particularly wet, in terms of kind of annual rainfall, and high nutrient environments. And it seems like once those lineages happen to land in these wet, high nutrient environments, that's when they kind of stopped there and they never progressed on to C4. So it could be that if you want to know what the kind of C2 land is, if you think of like C3 land being kind of like, wet and cooler, maybe dark, and then C4 land being bright, highlight and warm temperatures. If you think of what C2 land might be, where C2 plants are most thriving, if that answer is they do best in wet and high nutrient environments, that doesn't really make them ideal for an agricultural setting. I mean, you don't really want crops that you need to water and fertilize a lot. So I think we need to tease this apart, and we need to do some kind of manipulation experiments and understand exactly where C2 plants are going to thrive. That work still needs to be done. That's work that I was supposed to do as part of my Leverhulme early career fellowship, but then the lockdowns this past year have hampered it. But I'm planning to pick it back up in the coming months, and hopefully I'll have an answer to this by, well, I'd like to say summer, but that might be optimistic, but hopefully we'll understand it better soon. Okay, so that ecological breadth also comes down to this physiological flexibility. And this is something I alluded to earlier, how under non-photorespiratory conditions, C2 plants are going to look just like a C3 plant, right? There's no photorespiration. They're just doing Calvin cycle in the mesophyll. And under photorespiratory conditions, that's when it kicks in. That's when you start getting this more efficient C2 cycle running and recycling that photorespiratory CO2. So in that sense, C2 photosynthesis is environmentally inducible by nature. So these C2 plants have essentially C3 photosynthesis, and they tack on C2 on top of it, okay? And if you think of it that way, you can imagine that C2 plants, in theory, should do well in C3 environments, because they should be just as good as a C3 plant, right? In theory. They should do as well as in C4 environments, because when there's high photorespiration, they're going to recycle that CO2. So there's going to be, they're going to do okay in those environments as well. But they should also do particularly well in heterogeneous environments, because they can kind of turn on and off this C2 business depending on how much photorespiration there is. They should be able to do well in environments that are constantly shifting back and forth. So this is kind of the idea in theory. With that said, we do need to consider potential costs that might be associated with C2 photosynthesis and how that might play in here. But for the most part, you can see why these C2 plants are so ecologically widespread. It's because of this physiological flexibility, I hypothesize. And talking about this flexibility, another PhD student I wanted to give a shout out to, so this is Catherine Walsh. She's a first year PhD student, and she is working on rocket or arugula. And what we're trying to find out is if this physiological flexibility has consequences for crops. And what I mean by that is the only crop that uses C2 photosynthesis is arugula, right, is rocket. But what we know about rocket, if you talk to growers, it's incredibly frustrating to grow because it's very variable. It's notoriously variable. So the taste, the flavor profile will change season to season or probably week to week. It's hard for growers to know what they're going to get. And our hypothesis is some of that flavor variability, that inconsistency might come down to that C2 cycle and how much it's running. And we have reason to believe that it is linked. And so Catherine is doing some very, very cool work. She's trying to link the carbon, nitrogen, and sulfur metabolisms in C2 plants to understand how not only these metabolisms, but other metabolisms all interact to affect flavor. Because if we want to start introducing C2 photosynthesis into crops, we need to know if this is going to be a problem. The other thing she's looking at is arugula, or rocket. It's a hyperaccumulator of nitrate. And we need to know if that is linked to C2 physiology. And if it is, is it present in other C2 species? Because this could also be another important factor in if we do want to get this introduced into crops or not. We might have to consider that. Okay, so back to this physiological flexibility idea. It's not just that we have essentially a C3 cycle with a C2 cycle tacked on, but a lot of these plants also, like I said earlier, have this weak C4 cycle tacked on. So what you can have are plants that have multiple physiologies running potentially at the same time. And what the reason I'm bringing that up is, if you want to characterize the physiology of C2 plants, it gets very messy very quickly. And it's because it's just inconsistent. If you look at the literature, looking at things like water use efficiency and nitrogen use efficiency is kind of all over the map. And that's, I think, because you have these different physiologies running. And so to tease apart exactly what role C2 is playing, what this glycine shuttle is playing, you have to be able to separate it from other physiologies. So what I need to do, and one thing I'm planning to try to do, is to try to break down and understand the contribution of each of these potentially 3 cycles at any one point to determine its effect on C2, to determine the effect of C2 photosynthesis on these plants. But yeah, so I mean, I'd love to be able to say C2 plants have higher water use efficiency and nitrogen use efficiency. And if I cite the right studies, I can say that. But if you look more broadly, it's just a bit all over the map, if I'm being honest. Okay. So now I'm going to move, hopefully I'm not talking too quickly. Now I'm going to move on to the second part of my talk on engineering C2 photosynthesis for crop improvement. First, okay, why do I think C2 photosynthesis is worth our time? Why does it have potential to improve crops? And there's 2 main things that I support here. One is that C2 photosynthesis should, in theory, consistently outperform C3 plants. And 2, that C2 plants are inherently physiologically flexible. So I'll go on and talk about both of these in the next couple slides. So this first bit, this idea that C2 plants should consistently outperform C3, this comes from this recent modeling paper from Chandra Blasio and Graham Farquhar. This is 2019. They used a modeling approach and they said, okay, so if we engineer C2 photosynthesis into rice or C4 photosynthesis into rice, what would we expect? What kind of benefit would we expect from these engineering efforts? And in which environments would we expect the biggest benefit? I mean, without going into detail, they also engineered C2 plus C4 as well, but I'm going to keep it simple. And what they found is maybe unsurprising at first. So we'll go back up. And that is that, sure, if you engineer C4 photosynthesis into rice, you're going to see big gains, right? We know that there's a reason we're putting lots of money into this. But you're only going to find that in a small subset of environments, right? It's only at high temperatures and high light that you're really going to see the biggest, that amazing C4 benefit that we're aiming to get. The thing with C2 is you're not going to get as big of an improvement, but you're going to get it in almost every arrangement of environments. So small gains, but consistent gains is the idea. And to give you a bit more specifics of what they found in this paper, the C2 rice outperformed C3 rice under ambient CO2. It was a very small benefit though, but you get the biggest benefit of C2 rice under warm temperatures. So this over 35 degrees C, high light, 700 micromoles, not even that high light, and then low CO2. So that's when you're going to see the most C2 benefit. But also the benefit at warm temperatures with C2 photosynthesis is so strong that even under the higher CO2 you're going to see with climate change, C2 rice is still winning out. C2 rice is still worth our time, basically. So that's the outcome of this paper, is that it might be worth looking at C2 rice. So that is promising, considering I'm dedicating my life to it. Right. So, and the other reason gets back to this physiological flexibility. And I'm not going to go over it again, but the point is, if these plants really can do well in C3, C4, and heterogeneous environments, that's kind of ideal. Like if we can engineer crops that can turn on and off physiologies depending on what the weather is that day, that's ideal. I mean, that's kind of engineering crops for the future. That's engineering crops for an uncertain environment. And if we can do that in a way that's not costly, or that's not terribly costly, then that could actually be a really smart approach. And what we'd be doing there is we'd be potentially really expanding our crop regions. There could be areas that are not being used for crops that can now become used. And among other things, and I haven't mentioned this before, C2 plants are also notorious for growing on marginal lands. They for some reason do well in really poor environments, like poor soils and stuff. You know, if you look at the SAGE review papers, this keeps coming up. And it could be that engineering C2 photosynthesis into crops could mean we can start expanding agricultural lands into more marginal spaces. So this is what I'm hoping. These are the reasons I'm putting my time and effort into this C2 idea. So to summarize, the unique benefits of C2 photosynthesis are it's a CO2 recycling mechanism that boosts photosynthetic efficiency under warm, bright, and low CO2 conditions. This environmental inducibility allows plants to tolerate broad and variable environments. That's exactly what we're going to need in our current climate, but especially in our future climate with more climate change happening. And so that's what I'm getting at this last line. These crops will be primed for climate change, but also primed to fight malnutrition. And one of the reasons I'm bringing that up is because a lot of the efforts at the moment are to improve grain crops. And there's a lot of value in that. Obviously, we need calories, right? You need calories to feed people, but you also need nutrients. And so this C2 engineering program, I'm going to target some of these families like Brassicaceae and you know, other high nutrient families to get at malnutrition, which is another important issue in addition to just not having enough calories. Okay, so if we do want to go about engineering C2 plants, what kind of modifications do we need to do? And this part of my talk is coming from my recent Tansley Insight paper in New Phytologist. And I'll go through the few little steps that I propose. The first top is just the C3 phenotype. So this is kind of our starting point. And if you start with a C3 phenotype, the first step we need to do is prepare the bundle sheath. And what do I mean by that? I mean we need to enhance the organelles. We need more chloroplasts, mitochondria, and photosynthetic enzymes in the bundle sheath. We also need to improve that mesophyll to bundle sheath connectivity through, for example, more plasmodesmata or pit fields. And then finally, we might need to do some organelle repositioning along this in the inner centripetal wall. That's commonly touted as a requirement for C2 photosynthesis. I know for a fact that not all lineages do that. So I think that might be a lineage specific thing. So whether we have to do that in a crop engineering effort or not is still to be seen. We might be able to get away without that step, or it might be an optimization step. Maybe it'll work without it, but it'll work even better if we do find a way to move the organelles to the inner centripetal wall. Okay, so if we want to do this step one, luckily, there seems to be a way to do that. This Wang et al. Paper came out in 2017 in Current Biology. This is some really, really cool work that's come out of the C4 Rice engineering program. And what they found is that the constitutive expression of GLK, which is golden 2-like transcription factor, that leads to larger bundle sheath chloroplasts with 2 to 3 times more Rubisco and Rubisco activase enzymes. It creates larger bundle sheath mitochondria with GDC, and it increases the plasmodesmata junctions. So it seems like we can kind of prepare the bundle sheath with one step here, potentially. Okay. So what's the second step? After we prepare the bundle sheath, we actually need to functionalize the bundle sheath. And what I mean by that is we need to restrict GDC to the bundle sheath. It's one thing to have GDC present in the bundle sheath, but if it's also present in the mesophyll, you're never going to run the glycine shuttle. You need GDC to not be functional in the mesophyll in order to get glycine accumulating and cause the shuttle over to the bundle sheath. And it looks like, in my optimism, that we can do that in one step, right? So if you look at these two Adway papers, 2015 and 2019, there's something called the M-box region. And if you delete that, that causes bundle sheath specific expression of GDC, or specifically in these studies, it was the P subunit, which is the one that we seem to think is the most critical. So it could be that we could functionalize a bundle sheath in one easy peasy step, right? I'm going to go with that for now. And then the final step is we might need additional modifications to optimize the C2 shuttle. And, we will. What I think is that these two first steps with the GLK and the M-box deletion modifications, I think that should give us some kind of rudimentary C2 cycle in theory. Whether it works well, probably not, right? So there's probably going to be some additional steps we're going to need to do to make this work better. There's probably going to be other things we need to do. For example, we might need to enlarge the bundle sheath. But again, I think that's lineage specific because it'll depend on the C3 background on which you're trying to introduce C2. If you're working with a lineage that already has large bundle sheath cells, you're not going to need to put effort into enlarging them, right? So that's why I'm saying it's lineage specific. Okay, so what about the glaring thing that gets brought up a lot is what about gross leaf anatomy? And I think we don't need to make any changes to gross leaf anatomy to recreate a C2 phenotype. Recreating C4, one of the reasons it's been so tricky is that we need to introduce these additional veins, right? Because C4 photosynthesis notoriously has high vein density and few mesophyll cells between the veins, and that which occurs through high vein density. And that's been a bit tricky to figure out and to engineer, right? But C2 phenotypes don't seem to need that. And this paper that came out, I think it's about 2019, it's Ecology Letters. It was with Pascal Antoine and Colin Osborne. We just looked at one species. It's Allotrops simulata. And incidentally, you didn't think you were going to get away without with a talk with me not talking about allotropsis. I don't think I could humanly not talk about allotropsis in a talk, but here we are. This is a grass species that is incredibly unique in that the different populations kind of span the whole range of photosynthetic diversity between C3 and C4. You have C3, C2 type 1, C2 type 2, C4-like, and C4. And using that photosynthetic diversity, we can start understanding a lot more about what it means to be these different photosynthetic categories. And what we found is that the only gross anatomical difference between a C3 and a C2 plant is just the number of mesophyll cells between the veins. And in C2 plants, it was like slightly smaller, like slightly fewer mesophyll cells. But that was the only thing. That was the only difference between a C3 and a C2 phenotype in these plants. And yes, they had fewer mesophyll cells, but if you look at the literature at other grasses and the number of mesophyll cells between the veins, these C2 plants fell within the range of other C3 species, meaning that even though they had fewer in this study, it's not that that's different from C3 plants as a whole. So what I'm trying to get at and what I'm rambling on about is that I don't think there's any required anatomical modifications, gross anatomical modifications that we'll need. And if we can skip this step, that's going to be a huge boost to C2 photosynthesis engineering programs because it'll make it a lot easier. So that's currently where I'm standing. And I realize this is just one system. This is just one grass, and it could be different for other lineages. But it does give us hope that hopefully we won't need to do any kind of mass leaf rearranging. And overall, kind of the point I'm getting at here is that recreating a C2 phenotype in a C3 background is way easier than recreating a C4 one. So yeah, hopefully it won't be nearly as bad or as time and energy intensive as a C4 engineering efforts. Okay, so the last thing I wanted to point out here in terms of required modifications is it could be that there's going to be some interactions with other metabolic processes, like I mentioned before, that nitrogen imbalance issue that we might need to address. So it could be that we have some kind of sticky metabolic stuff we might have to think about in the future. What those are, we don't know yet, but I just wanted to flag it as something that we'll need to be paying attention to as we start engineering this C2 cycle. Okay, so I'm going to go, I think I have about 5 to 7 minutes. I'm going to go over my crop engineering program and what I'm planning to do. And the first step was picking crops that I want to work with. So I am targeting 4 plant families to work on. And I picked these specifically because they're crops that are important crops, but they're also in families where we know C2 photosynthesis has evolved in the past. And that's important because it might be that these crops are already kind of predisposed or maybe enabled, if you will, to take on a C2 phenotype more readily than families that never evolved C2 or C4 photosynthesis. So that's my thinking. That's kind of why I'm picking some of these crop families that I am. And the families I'm working with are Amaranthacea, so that includes crops like quinoa and sugar beet, also spinach. Asteraceae, so this is a sunflower and lettuces. Then there's the Brassicaceae, so that's all our cruciferous vegetables and things like oilseed rape. And then finally the Poaceae, the grains, so things like rice and wheat. All right, and so thinking of these 4 families, the first thing I want to do is understand their phenotype. If I want to recreate a C2 phenotype, I need to know exactly what I mean by that, right? So the first step I'm going to do is look very closely at the different C2 plants in these 4 families and look at their, exactly what their gross leaf anatomy looks like. We'll be doing ultrastructure and immunolocalizations to really make sure we know exactly what we're trying to recreate. The next step is understanding the gene expression profile. So we're going to be using this new fancy technique to map the spatial gene expression profiles of C2 plants. And we will be doing it also with close C3 and C4 relatives to put it into a larger context. And to do this, we'll be using this 10x Genomics Visium platform. And that should tell us exactly which genes are being expressed in which cell types and how that maybe differs across the leaf. And so, yeah, certainly within the different cell types. So that's the main thing we're trying to do there. And once we have this understanding of within the 4 families, the C2 phenotype and the C2 genotype patterning, then the next step is, of course, to start the engineering work, right? I'm going to try to recreate C2 photosynthesis in these crops. And to do that, I'm going to go to this first, what I was talking to before, this kind of 1-2 punch, the GLK, constitutive expression and the M-box deletion. And that, I think, will create a rudimentary C2 cycle. So I'll start with that, with the GLK and M-box work. And then I'm going to add on anything we learn from the phenotyping and genotyping patterning work to kind of optimize those systems, ideally. And so by the end of the first part of my fellowship, I should have, in theory, and if all the stars align, a working and hopefully optimized C2 system working in, I'll be working first in camelina and broccoli. I'll be doing those kind of in parallel, working with the folks at the John Innes Center. And then once I understand and get that working, I want to run a whole cycle from the beginning to optimization fully in oilseed rape. So that is my initial approach, is to focus on Brassicaceae crops, take what we know from literature, add some new information that I don't think anyone's done before at this level, and hopefully be able to recreate that in these C3 brassica crops. And then, yeah, so I'm going to take everything I've learned from these first 3 steps, the phenotyping, genotyping, and engineering work, and I'm going to create these engineering instruction packages. And so I want to know in those 4 families, if you want to start additional C2 engineering programs, I want to know exactly what to do, what worked, what didn't work, what do we know about the phenotypes and genotypes. So hopefully that can be a useful resource for my future projects and other people's future projects. And just to wrap this up, the last few steps, I of course need to field trials. That will be done, the Nebraska work in the US in collaboration with the RIPE, Realizing Increased Photosynthetic Diversity or Efficiency program, in Argentina with the University of Argentina, Buenos Aires, and in Canada with our partners at AgQuest. And then the next step in my optimism will be to bring these C2 crops to market and to work on some commercialization work towards the end of my fellowship. And then, the last step is to start all over again. My plan is to take what we know from Brassicaceae, see if we can get that to work, and then start all over again in some Asteraceae and Amaranthaceae crops. And to do that, I'll look to these kind of engineering packages I've already put together. And hopefully, I'm not starting at the ground floor, but I'll already have an idea of what I need to do and how I need to do it. So the outcome of all this should be that I will have created the world's first C2 crop engineering program. These crops should have incredibly broad environmental tolerance, and they should improve yield in high nutrient crops, which should target malnutrition. And before I finish up, I want to say that we are hiring. I have 2 senior postdoc positions. One is this really cool phenotyping work that also includes a spatial transcriptomics that you will be trained on. So don't worry about that if you've not done it before. That position is open now. Applications are due 15 May at this point, and it's a 42-month position, and it does come with Tier 1 Global Talent Visa support if you are international. The other postdoc position is in all the biotechnology work, so the Brassica transformations. This is a 36-month position, and again, it has full visa support. So international students are welcome. And then there's also a part-time lab technician post without visa support, unfortunately. But point is, we're hiring, and if you're interested in this work, do consider coming to the Lundgren Lab if you're looking for work in the near future. So finally, I'd like to thank the Lancaster photosynthesis team, everyone involved with that, then Colin Osborne and Pascal Antoine Christin at the University of Sheffield, my PhD students, Sophie Young and Catherine Walsh. And finally, the newest member of the Lundgren Lab is a grass enthusiast. And my funding comes from UKRI and the Leave of Human Trust. Thank you very much.